we want to talk about the most amazing elaborate antennae of some of the Lepidoptera. So you can see on the left here some contrast between female and male antennae of the same moth species, noting that in this particular case, the males have far more elaborate sensory apparatus than the females. And we'll talk a little bit more about this. And you can almost predict that in a species like here, that it is the males that find the females rather than the other way around. And so in this case, you can probably assume more often than not that the presence of highly structured, fine antennae is associated with location of mates. And in this case, it is almost certainly that it's the female that produces the pheromone to which the males are attracted. And you can see here in the contrast to butterflies' antennae, where structurally they're not as complex or finely structured, but it's not to say they are less sensitive. They're maybe just sensitive to different things. And you can also probably predict that one of the reasons why a butterfly's antennae is such is that the reliance on visual cues in addition to chemical cues and olfactory cues in the context of being a butterfly, which typically tend to be day flying, active in the day type animals. Whereas in the case of moths, you typically associate them with being active at night, and so they need an apparatus that sort of equivalently suits the lack of a visual stimulus. So most moth antennae, they are among the most sensitive chemical detection organs known in insects. They're capable of detecting trace amounts of odorant molecules. Sometimes, even a handful of pheromone molecules in the air is sufficient for them to detect something, right? And it's this extraordinary sensitivity achieved because of how densely packed and densely covered these sensory hairs are on the antennae, right? And each sensory hair is its own little olfactory receptor factory, right? Where it's, all the ORNs are concentrated and each finely tuned to very, very specific chemicals. And so in many species, I mentioned a while ago, the male moths have this far more elaborate feathery, what we call bipectinate. We'll talk about that in the next slide, compared to the females, because in those species, the males are the ones that are relying on sex pheromones released by females to locate them over, what is typically vast distances. And so you can see here in some scanning electron microscopes slides, micrographs of the antennal condition in moths. So bipectinate is this phenomenon where you have a branched sort of structure. So that's very obvious in the photo we saw a while ago of the male moth's antennae. You can see here that there is diversity in the way that this bipectinate condition can exhibit itself. Here we can describe it as something strongly bipectinate, and also you can describe it as weakly bipectinate. And of course, there's probably evolutionary drivers as to why one is favored over the other. And you can see here whether it's the lateral or the ventral view that you can see fine structures upon fine structures. So surface area is exceedingly high to house a huge density and huge diversity of ORNs, right? I also wanted in this video to sort of also show you the flip side of this bipectinate condition, right? The word bipectinate is a descriptor to describe the physical condition of these structures. So while on one end it could be in the antennae at the top of the insect, it could also very reasonably be on the other end. And so I wanted to show you this absolutely spectacular moth that you see here on the left, which we have in parts of Southeast Asia and also in Northern Australia. But something a bit closer to home on the right, you see the common crow butterfly, Euploia corae. And you see that at the end of their abdomen, the projection of these, what we call hair pencils, or more technically, we call them corymata. And these can, of course, be retracted and everted as required. And in a weird way, it's kind of like the reverse. And you notice that in the case of the moth on the left, the presence of these highly elaborate corymata is correlated to a very, very simple antennae. And so that tells you in this particular species that there's a very, very good chance where the roles are reversed, and it is instead the males that release pheromones that bring females in. So the male antennae in this species is not very elaborate because he does not need to look for females. The females are coming to him. But in order to get the females, he's invested in a different strategy that comes on the other end, which they invert these hair pencils. These are specialized retractable tufts of elongated hairs and scales, mainly in male insects, to release those pheromones, right? So these structures are associated with pheromone storage and release. They don't actually detect anything. They're not receptors. They are emitters, right? And during courtship, in the case of Euploia cori, the common crow, it's been suggested, and this is work from Queensland researchers, it's been suggested that frequently the male sort of flies just above the female, and then everts his hair pencils to release like sort of the volatiles that then sort of in a weird way get sprayed onto the female, which causes her to arrest and land, and then the male can actually approach her, and then a series of behaviors will ultimately lead to reproduction. But yeah, it's really cool that the same sort of condition is exhibited whichever strategy you go with, whether it's a male to female or female to male. That the solution is always to invest in a structure that is bipectinate and increasing surface area to either emit or to receive signals.